Warm welcome to everybody joining us today for today's ISNTD Connect session. We have some very special guests here today from Merck, Dr. Beatrice Greco, from Find DX, Dr. Sarah Nogaro, and from Janssen, part of the Johnson & Johnson family of companies, Dr. Lieven, Stuyver there. Hello to everybody and welcome. To all of the audience that are out there, we've got a lot of participants joining here today. At the moment, it's showing up on my system at about 250, but it's rising as we're speaking. I'd like to say a warm welcome to everybody, and also to ask everybody if you could just give a shout out on the chat in terms of your name, organization, and country. That would be great. It's very exciting times here, not just because it's the beginning of the year, but also because this is part of our celebration of the World NTD Day, which is launched on the 30th of January in 4 days from now. And prior to that, there's going to be the official launch of the WHO roadmap, the NTD roadmap to 2030, with all the guidelines for elimination and control and the various strategic viewpoints shaping the engagement of all actors, all players in this particular space in terms of the NTD world, the global health initiatives around NTDs. And really, that report, it was ratified back in April by member states, but it was on the back of a long consensus and collaborative framework put together by the WHO. So there's not a prescriptive kind of guideline. It's really a synthesis of all the ideas and all the inputs from all the actors involved in this space. So it's quite a unique kind of set of guidelines and very reflective of the approaches, whether that's preventative or curative in NTDs. I just wanted to quickly share some of the strategic shifts in that. Okay, so the roadmap itself, once you strip all of the guidelines down, there are three major shifts that really are taking place in the WHO's guidelines. One is for accountability in terms of impact, measures around impact. The other shift is really in terms of the pragmatic approaches taken by this ecosystem. And the third major shift is in terms of ownership, program ownership. And if you open these up a little bit, if I probably have to move myself from that top corner, you'll see that the first shift, historically, we've had a process-driven approach. Success within those kind of programs have been measured really by actions taken. And the shift is really towards an impact-orientated approach, i.e. kind of measuring the public health impact of NTD interventions, whether that's drug-based, whether that's preventative measures, diagnostics, any of the disease surveillance things. We're looking at what is the actual impact to those communities at large using endpoints around public health. So that's the first shift. The second shift, as I mentioned before, is more in terms of pragmatic approaches. And from that, really, we've all heard the kind of rhetoric, as it were, really we've all heard the terms, at least multidisciplinaryism, multi-sector approaches, cross-cutting approaches, but this is really one of the most exciting shifts, in my opinion, because it really is trying to leverage, movement from a disease-specific silo, a vertical kind of approach. Those approaches are very limited in terms of where they touch on the national healthcare systems in countries, as well as the other sectors. Those sectors are, include technologies, platforms, different approaches which wouldn't normally be thought of when it came to a specific disease, but were being used in different settings. This shift to a more cross-cutting approach, integration across NTDs, almost mainstreaming into the national health systems that are present, and then a sense of coordination between adjacent sectors. They're really there to strengthen country capacity and build a global support around us. It's a very, very exciting shift in that sense. In terms of the kind of third shift, program ownership, the WHO are really pushing for movement from the kind of traditional externally led, I've put agenda, but externally led, expert-led kind of support, all the way from support to donor funding to a more country sense of ownership, a sovereign sense of ownership. Within that is included political will, also financing, integrating to national health plans, budgets, and then supported by partners and donors really to kind of help overcome all the challenges there. So they're the three shifts. We were really excited by today's webinar. As some of you may have seen, we had a series from the Global Schisto Alliance all of last year, looking at various aspects around schistosomiasis from their perspective. This particular, and that's continuing this year, this is the first in the series we're doing with Merck. They obviously, as you well know, are very much involved in the field of schistosomiasis with their drug praziquantel and the reformulation efforts around that. And we really wanted to kind of frame all of our Connect series around these three major shifts. And this really, for us, represents one of the first donor-to-donor collaborations between the Janssen Group and Merck, of its type. And so we're very excited to have this opportunity to bring this out into the wider domain. And so, without further ado, I think that's enough from myself. I think it's time to hand over to Dr. Beatrice Greco from the Merck Global Health Group. And so I think a round of applause for Beatrice. And if I could ask the other presenters, Dr. Stuyver and Dr. Nogaro, just to kind of take themselves off the camera for a second and then come in, that would be very much appreciated. Over to you, Beatrice. Thank you.
Thank you very much, Cameron. And also thank you for having us and hosting us in this ISNTD presentations. So good afternoon to all. Today, what we would like to do with this first presentation is first to present to you the type of work that we conduct at Merck together with our partners, with the intent on supporting the community to reach elimination of schistosomiasis by 2030. First, I will just give you just a very few words on who we are. So we are Merck, a worldwide company founded more than 350 years ago. And our goal and our mission, our purpose is to deliver science and technologies to significantly impact human lives. And this through actually our three major businesses, core businesses. Healthcare, it is delivering innovative treatments for patients suffering from oncological and immunological diseases. Life science, that is through its very large catalog of various products and solutions, is empowering the overall scientific community. And performance material, that is developing high technologies in a sustainable manner that is impacting our everyday lives. We are known as Merck throughout the world, but we are known in the United States and Canada as EMD Serrano, Millipore Sigma, as well as EMD Performance Material there. When it comes to the Global Health Department at Merck, our goal is to improve the health of underserved population in low-middle-income countries through combining our knowledge, our competencies with the ones of our partners, and to deliver innovative solutions. Our strategic intent and our key focus has been really to look at some specific non-communicable diseases and to prevent them and control them in low-middle-income countries. To look at also aspects of malaria from the prevention perspective in particular, and very much focusing our attention on eliminating schistosomiasis as a public health problem. Just a few words on schistosomiasis. I'm certain that among the assistants there's many experts, so bear with me on the simplicity that I'm bringing here. So schistosomiasis is one of the 20 NTDs, neglected tropical disease. This is a parasitic disease that is acute and chronic and is caused by the exposure of humans to waters containing the larval form of schistosoma. Once the infection has occurred, then there is maturation to adult worms, and then there's secretion and excretion of eggs. Eggs that then are going to be excreted either via urines and feces, maintaining the life cycle of transmission of the parasite, or the eggs are being trapped within the body of the human host. And this is leading to very severe morbidities, cancers, fibrotic tissues, anemia, cognitive dysfunctions. And what we do see is that actually schistosomiasis is endemic into 78 countries, primarily within the African continent. More than 200 million people are requiring treatment. As I was mentioning, the severe morbidities are leading to a certain degree of death, and a lot of and numerous disability burden in populations. WHO NTD roadmap, as Cameron was mentioning, the new NTD roadmap 2130 is going to be issued and launched officially in about 2 days. It's giving guidance to the community as what are the elements, what are the targets and the elements needed to reach elimination of about 20 neglected tropical diseases. When it comes to schistosomiasis, the goal is elimination as a public health issue, which has been defined as below 1% of prevalence of heavy intensity infection. Of course, there are targets for a number of countries reaching elimination by when, but ultimately by 2030, 78 out of 78 countries should be at the point of eliminating schistosomiasis as a public health issue. This comes together with a number of actions, such as, for example, strengthening the implementation of existing interventions, developing new tools, strengthening the cross-collaboration between different sectors, including WASH in particular, but also ensuring that there are the proper resources and access to the tools that are needed. So an integrated approach in a way. An integrated approach that at Merck we have been deploying and using in our approach to fight schistosomiasis for the last few years. And this integrated approach highlighting also a certain number of strategic intent. The first strategic intent for our approach has been to support country elimination through providing existing targets, addressing key medical product needs by focusing on the development of innovative tools, fostering cross-sectoral partnership really to trigger resource and cross-disease efficiencies. We are in a field where resources are tight. We need to create and to leverage on those resources. But all this works only if the health system in which we are deploying those new tools, those medications, is resilient and strong. So we are looking also at ways to strengthen healthcare systems by, in particular, building local capacity and knowledge also. And last and not least, it's really addressing the question of fostering access to those schistosomiasis medical products. And that is going to be really the key topic of today's conversation with the various presentations we are going to be giving. Now, more concretely, how do we translate this strategic intent, this integrated approach? Well, this is translated by a series of interlinked programs going from a donation program to advocacy activities, R&D, as well as educational programs. And what I'm going to do now is to give you a few highlights on those various projects. Of course, those various projects are associated to the key pillars to reach elimination: drugs, diagnostic, WASH, health education, but also addressing our own strategic intent. The first one being reaching country elimination by providing existing treatment. Merck has been donating praziquantel, which is the treatment against schistosomiasis, since 2007 through a partnership with WHO. We are up to 200 million tablets delivered per year, and we've celebrated the 1 billion tablet provided a couple of years ago of praziquantel since the beginning of the program. And we are very happy that we've already reached over 500 million treatment of school-aged children through 47 African countries, and the commitment continues. So this is one element. Beyond this, we want also, as I said, to address the key medical product needs for supporting this elimination agenda. And one element that comes is the need for effective interventions and new alternatives to complement praziquantel in particular. So this is the work that we conduct through our drug discovery platform, where we are looking at delivering new anti-schistosomal drug candidates to address key gaps in the target product profile that is needed to really complement praziquantel. And this has to do with resistance, but also ensuring that the new drugs are going to be targeting juvenile form of the worm. As mentioned, a pediatric formulation of praziquantel is needed. This is a need that has been highlighted for now a couple of years. And through a consortium that was launched already in 2012, this is what this product development partnership is aiming at by developing a new oral dispersible tablet of the praziquantel for children below 6 years old. And today we are in phase 3. When it comes to diagnostics, very clearly we need to have and we need to develop sensitive point-of-care diagnostics. This is the work that we are currently conducting with FIND and on the development of a rapid diagnostic test. FIND and partners, and Sarah will be presenting to you this exciting development that we're conducting together. But also highlighting some of the key elements that we are looking at more from an access perspective. We are also looking at improving what is existing. What is existing today is cattle cats. We're measuring eggs, counting eggs. And this is a work that we are currently conducting with Janssen on developing the next generation of microscopic assessment on an artificial intelligence-based approach for detecting co-infection of schistosomiasis and soil-transmitted Helminthiasis together, including a monitoring and evaluation plan. And actually, this is leading us to our other pillar, which is how to trigger and how to integrate with various sectors, and in particular to trigger resource but also cross-disease efficiencies. And here the clear example of this work with Janssen is an example of cross-donor collaboration to address not only the elimination agenda of schistosomiasis, but schistosomiasis together with soil-transmitted Helminthiasis. And Lieven will be presenting to you in a little while the elements of this work, looking at STH also, but thinking already beyond those two indications. When it comes to also cross-sectoral partnership, it's very clear that WASH plays a key role in the elimination of schistosomiasis. And here we have been developing with a number of partners also concepts and programs on development of water technologies and platforms to remove the most vulnerable populations from being exposed to those waters containing the parasite. But by doing so, what we do also is indeed looking at limiting exposure to waterborne disease in a larger setting. And when you know and when we know that actually waterborne disease is one of the factors leading to a major cause of death in children below 5 years old. This is the type of program that we are really looking forward to see an impact on both our own disease of interest, schistosomiasis, but also looking at how this is also impacting survival of infants in some of those regions of where we are going to be deploying this program. Again, cross-disease efficiency, as I mentioned, schistosomiasis has many morbidities. One of them that is has been, again, somewhat neglected among this neglected disease is a genital form of schistosomiasis, and more specifically in women. And because this is very much associated, now there's increasing evidence, and this is increasingly associated with various types of cancers, but also HIV. So further understanding female genital schistosomiasis may lead also to further understanding of some aspect of HIV transmission, as well as approaching the question of cancer and assessing data and information around cancer and cervical cancer in particular within this type of populations. When now we come to the fourth pillar, as I said, to lead to elimination, it requires to have a very strong health system. And strengthening health system is part of our strategy for this particular pillar of schistosomiasis, but also for the larger sets of information and type of activities that we're conducting for malaria, for example. And here, the way we are approaching the question is by really looking at building local capacity and knowledge. Local capacity, again, strengthening health system through the WASH approach. It's not about just removing and trying to remove the exposure of people into contaminated waters, but it's also to look at where are patients going to go to get treated. And this is at the level of healthcare centers. And here we have identified very critical gap in terms of access to water and to clean water in a sustainable manner in healthcare centers. I believe with COVID, this has come back very clearly in our minds that having access to basic clean water to wash your hands, to wash your face is the first barrier against a number of infectious diseases. And in the context of schistosomiasis, this is also the case. Patients suffering schistosomiasis will go to healthcare centers. They will need to have access to a more holistic approach of not just drugs and potentially diagnostic, but also being cared for normally and within a setting of health system that is strong. So access to water in healthcare centers. Another way of building local capacity for us is to look more specifically within the programs that we have in our pipeline, and in particular, an example is the pediatric praziquantel formulation, where to ensure access, one key element that we're looking at is actually tech transfer of capacity for manufacturing in low-middle-income countries. Finally, building local capacity also from a knowledge perspective rather than an infrastructure perspective. It's really the type of activities that we conduct from a health education perspective with a number of partners, but also the tight and very strong collaboration with the Global Schisto Alliance, where there is really a lot about accelerating progress in knowledge around schistosomiasis and how to tackle the elimination pillars that are needed. And finally, this leads me to the last pillar of our strategy, which is really fostering access to schistosomiasis medical innovation. And here we will talk more about this piece in the next couple of talks, but for us really this is the key gap. We are talking about really an uncharted territory. From an advocacy and funding perspective, we are conducting a number of activities together with the Uniting Effort for Innovation, Access and Delivery platform, And here actually there's 2 examples of publications that came out last year, and I would recommend you to get to them because they are quite informative on doing the landscape analysis of the elements that are missing for access in general, and starting to think with the community about what are elements on which we can start working on. But more and beyond the advocacy element, there's really a need now to think through what are the concrete elements on which we need to work to ensure access to the pediatric formulation as well as access to the diagnostics when they're going to be available. From the pediatric praziquantel formulation perspective, you will hear, and this will be launched officially in a couple of months, we have been awarded a co-funding from EDCTP and GHIT on what we call the ADOPT program to assess those various elements and to put forward a program to really test an access strategy for this formulation. But from a diagnostic perspective, I think you will hear from Sarah and Lieven that there's also a lot of open questions, and even if we do have an ADOPT program, there's a lot of questions pending on how do we do this really concretely. I just want to finish by saying that this integrated approach with multi-type of programs going from donation, advocacy, R&D, education, fostering cross-sectoral partnership to leverage the resources and strengthening the health systems. All this work is actually an activity that where we have a grip on it, where there has been a lot of progressions in the R&D space, as well as the implementation space. But what we see is that really this access piece is the key next gap that we all have to face together. And also I want to mention that all this work cannot be done alone. And we're extremely fortunate to have many talented partners around us with whom we are working to deliver on those various programs. And those are from non-for-profit organizations, academics, profit and private companies also, but also countries. And the last but not the least, also co-founders, such as the Unfound, GHIT, and EDCT. So with this, I'd like to stop and back to you, Lieven, and Sarah and Cameron. And I will stop here.
Thank you very much, Beatrice, for that. What a wonderful presentation, and what a level of commitment. 520 million treatments have gone out to people across 47 countries. I think the WHO lists 52 countries in terms of endemic for schisto. So what an achievement in its own right. And then on top of that, this real focus on access, it really is going to spell out the next 5 to 10 years in terms of a pivot point around all of the engagement that everybody's really doing. So we're really thrilled to hear that. Very happy to hear about this Uniting Efforts arrangement as well, because as you know, we had the GHIT Fund webinar before the Christmas break. And in fact, we're talking to them as well about maybe downstream of this, a webinar looking at that with the government of Japan and GHIT and their partners on that. It's such an important point, and it's so encouraging to see that level of commitment move from the obvious the donations into this real life setting in terms of addressing the access barriers, the issues around that. So a round of applause, a digital round of applause for that, and thank you very much for sharing that with us. I think before we go to the Sarah's, I just want to give a quick shout out to some of the people. Dr. Carlos Texiria from Brazil, Dr. Ingi Krodil from Germany, Dr. Poppy Lamberton from Glasgow, Professor Bonnie Webster from London. We've got Dr. Francis Kimengo Francis from Cameroon. We've got Dr. Bartonek, Zygmunt Bartonek from Prague, from Cambodia, Dr. Boran Huat. So we're getting a lot of Anouk Gras Global Schisto Alliance. We've got such a wide array of people coming in terms of joining the call. Dr. Uliata from the Ministry of Health in Jakarta, Steffi Norp from Freiburg in Germany, Spain, I could go on, Aidan Emery from the Natural History Museum in London. So many names that we know, and new kind of attendees in terms of the geographic spread. I think that's wonderful, and it's really kind of a testament to not just the subject matter, but also, as you've shown, this real desire for collaboration, and it really plays to that, the second of the shifts in the WHO roadmap in terms of this movement from just a vertical approach to bringing in, practices, platforms, technologies that are going to impact that particular disease setting from different. You mentioned WASH as an example. That was wonderful. So thank you very much for that. And I'm sure we're already getting questions coming through on the chat box. So I'm sure you'll have quite a lively Q&A at the end of all of this. But thank you very much for that, Beatrice. As head of R&D at the Merck Global Health Institute, I'm sure you're a very busy person, so we really appreciate you giving us time for that. I think we'll move on to the next presentation. As Beatrice had mentioned, they're working with FIND, one of the major PDPs, or the great success story we often hear about, the DNDi, in terms of the therapeutic PDB that's really done a lot in terms of getting novel drugs out there. But we very, very rarely, I think, personally, or at least not enough, hear about the diagnostics world. And certainly it's a major gap that needs to be addressed moving forward. And the biggest success story in all of the diagnostic story in terms of NTDs has been FIND and the work that they're involved in from their Geneva offices worldwide. So we're very, very happy, very honored to be joined by Dr. Sarah Nogaro, the Senior Scientific Officer for NTDs at FIND, and to talk about schistosomiasis, rapid RDTs, rapid diagnostic tests to support control programs in monitoring treatment impact and reassessment mapping. I'm going to hand you over to Sarah, and I'm going to just come off the screen, and if I can ask Beatrice and Lieven to do the same, that would be brilliant. Over to you, Sarah. Thank you.
Thank you very much, Cameron. Good afternoon, everyone. So my presentation today will be about the work that FIND and its partners are doing in developing a new rapid diagnostic schistosomiasis diagnostic test to support national control programs to monitor the impact of their treatments, as well as for reassessment mapping. So somewhere on this line sits, on the x-axis, sits national control programs for schisto. And at each stage, there are specific diagnostic needs, and you will hear more from my fellow presenter, Lieven, on this later on. Now, the majority of the work that has been done in the red zone has been done using microscopy-based methods, CATs and urine filtration. Yet, as we move towards the right of the triangle, towards the green, that's towards elimination, there will be new diagnostic methods that are needed, that the ones that we've used until now in their current formats will no longer be useful. Most of you probably in the audience are aware of the advantages and disadvantages of the current diagnostic methods used for schistosomiasis, based on which we provide treatment, or ministries of health will give treatment. I'll go through these very quickly. The advantages are they're highly specific for the disease that we're looking at. They're field deployable, easy to use. They provide quantitative results. But also, not only are you looking for schistosomiasis eggs in the stools, you're also looking for soil-transmitted Helminths. Unfortunately, though, the microscopy-based methods, they lack sensitivity. They're time-consuming. You have to read multiple slides, collect samples over multiple days, and also juvenile infections cannot be detected. So what this means is that currently microscopy underestimates true prevalence, which can lead to premature stoppage of MDA programs, especially when the intensity of infections are very low. And by stopping prematurely, it leads to recurrence of infection to original levels soon after. However, as Lieven will show in his presentation, there are current methods to try to improve this microscopy tools as well. So FIND and its partners are developing an easy-to-use, accurate and affordable schistosomiasis rapid diagnostic test, which is looking to detect circulating anodic antigen, the CAA. CAA is continuously secreted by living schistosomes, both juvenile and adult worms, and can be detected in the blood as well as the urine, and has comparable sensitivity to repeated microscopy. For this RDT, there will be no sample prep required, nor a reader for detection. And the intended use case is really to support the national control programs to monitor the impact of their treatment campaigns, but also to be used in reassessment mapping. To give it a bit of context, the FIND project on schistosomiasis started in 2018. And at the time, it started with 4 partners. And over time, this grew, both in terms of key schisto opinion leaders, research institutions, funders, and national control programs of endemic countries. We are now 8 partners involved in the consortium, and shown here on the map where we are, where we're working and with whom we're working. To give you a bit of a context of the project and the work that we've done since the start of the project, the first part is really about defining the target product profiles, the TPP. That's a document that is required by the test developer company, where it outlines all the attributes of the tests that is then used to produce the RDT or the diagnostic test that you want. So the TPP was developed, and then it was endorsed by the WHO Working Group on Guidelines for Monitoring and Evaluation and Reassessment Mapping back at the end of 2018. And based on that, on the green line, the development of the CAA RDT started. And the first sort of prototype was available in November 2019, where it was field tested in Kenya on a small subset of individuals. At the same time, around that time, there was the new group, the Diagnostic Technical Sorry, I got disconnected. Hopefully you can hear me back. At the same time, in November 2019, there was a new advisory group to the WHO NTD department, the DTAG. Now, the role of the DTAG was actually to set out TPPs for different diseases and identify the main use cases for those diseases. So the first meeting was in October 2019, and then they formed different subgroups for each diseases or group of diseases. So the Schisto DTAG subgroup was formed in April 2020, and we were tasked to do a diagnostic landscape of current existing diagnostic tools, as well as identify the priority use cases for schistosomiasis. And based on that, we developed a draft of TPPs, which are tool agnostic and biomarker agnostic. And then they are currently under review with the main DTAG working group, as well as the WHO NTD department, before they will be publicly available for online consultation and for feedback from external review. So whilst this is going on, there is a development of the RDT. So after the alpha testing that was done in Kenya, the results from that study, as well as other studies that were continuing in the lab, the test developer continued to increase the performance of the prototype and created a version 1.2, for which the performance was verified in a lab-based setting. Now the prototypes have been shipped to the field study sites, which I will explain in a little bit, But in the meantime, we're also doing stability testing. And with R&D activities, it's also very important to look at the access component. And so we started doing some activities back in 2009 for the investment case, for which some of you who are probably attending the call today were interviewed, and to get some feedback on the schisto RDT. So where are we now and where are we going? So currently the field evaluations have started in Kenya and in the Philippines. At the same time, there are some more fundamental research questions that need to be undertaken. And the results from both of these pieces of work will inform the further optimization required for the prototype. It will then be lab validated and optimized with a further smaller field evaluation before it goes into design lock, transfer to manufacturing, and validation verification studies in the laboratory setting to then move into the clinical validation trials. But all of this going on, we will start working in depth in the access activities in partnership with Merck, but also other partners. So where are we now? So this is the kit in which the prototype comes in. So you've got a chase buffer bottle. You've got the actual test device where you've got a sample well, and then the reading frame where you can read out the results. And then you've got a scorecard where you can score the intensity of your test band with this scorecard. How it works, you have to use 50 microliters of finger prick blood or venous blood. You apply it into the sample well. You wait for 5 minutes for it to be absorbed. After that, you'll add 1 drop of chase buffer and wait for 15 minutes. And then you've got your results here. So the first 3 tests are positive with CAA present, are able to be detected. And then you've got a negative one here. So some results from the lab where the performance of the prototype was verified and actually met the TPP requirements. So here you've got the standard curve of spiked adult worm antigen, of which CAA is part of it, in naive blood. And what we were very excited to see, that at the 30 picogram per ml of CAA, we were able to detect a signal, which is the limit of detection for the test. What we also did was to compare the performance of the test with a very highly sensitive lab-based assay, which is the UCP-LF-CAA assay, which is based out of Leiden University. Now, the antibodies that are used in the UCP-LF-CAA assay are the same that are used in the prototype that we are developing at FIND with its partners. The UCP-LF assay requires sample prep and a reader for detection, and it's fully quantitative. So what we were really proud of is the very good correlation between the testing of the samples with both assays. So that was very encouraging. So the field evaluations have started now in the country, so both in Kenya and the Philippines. So in Kenya, we will be going to 2 schisto endemic sites, one that is for Mansenai and the other one for hematobium. And then we'll be testing in a non-endemic area for specificity. In the Philippines, one of the schisto-endemic sites will be for Japonicum, and we will also be testing for a non-endemic site for specificity again. So the study design, we will be collecting stool and urine samples for 3 consecutive days. And also on day 1, we will be testing venous blood and finger prick blood on the prototype RDT. Now, for stool and urine, we will be doing the CATOCATs and urine filtration, as well as PCR on day 1 samples only. And for the bloods, so on day 1, we will be doing the testing on the prototype RDT, and then an aliquot of all the blood samples will be sent to Leiden University to be tested by the UCP-LF assay, as well as aliquots from the urine as well. And a subset of RDTs will be reread 24 hours later as it is one of the key attributes of the TPP for stability of the results. So finally, the question on access. And here it's really important to think about access as we start to develop as we start developing the test. What we don't want is that we have a test that is ready to be used and available, but actually there is no access plan. How is it going to be rolled out? Who's going to buy it? So we need to start thinking about this as soon as we start developing the tool. So whilst access is a term that encompasses many meanings, perhaps one of the key questions we need to ask ourselves is, how will the new tool be rolled out? What are the market barriers for such a tool, and how should we address them? So back in 2019, FIND did an investment case to evaluate and quantify the market potential for the launch of a schisto RDT in schisto-endemic countries. We did this by a range of expert interviews, collecting both quantitative and also we did qualitative data. And we wanted to look at, quantify the market need and the market demand in 30 countries. And actually some of you who are on the webinar today were probably solicited for this work. And what I'd like to share with you on this slide is a few highlights from this work. And what are some of the barriers we need to address as a community? So I'll give you four examples. The first one that was highlighted was around policy. So WHO guidelines for schisto are seen as the highest normative authority, but are not always strictly adhered to by countries, either by lack of clear guidelines or the lack of financial resources. So the question we should ask is, how will these guidelines around reassessment mapping and M&E evolve as we move forward? The next point is around funding. The majority of funding for schisto goes towards treatments, and only very little goes towards diagnostics. But from that, actually even a smaller proportion goes towards access and access activities. In addition, to ensure future sustainability of programs, we need to ensure domestic funding. And in this context for access, we're referring to the pathway to commercialization, which includes gathering evidence which you would obtain from conducting clinical trials, which is part of the data that is required as part of the regulatory dossier and registration dossiers that you need to submit. But not only are we looking at the pathway to commercialization, but we're also looking for post-commercialization, so post-marketing monitoring, which checks for the adoption of the tests. So the question here we should be asking is, how is funding landscape going to evolve in the future? The next point I'd like to raise that came up was around the registration process. National registration process and LMICs are complex and highly fragmented. All countries are different, each having different specific requirements. So how can we harmonize the registration process across countries? My final point will be, or my final example of topics that we need to think about in terms of access, is around procurement and the distribution channels. There are limited players for procuring new tools, and the distribution channels are unclear. So what is needed to identify the commercial partner and then look at who and how the test will be procured and distributed? So thank you very much, and Cameron, I'll hand over back to you.
Thank you very much for that, Sarah, and what a great presentation. That final slide really illustrating the white elephants in the room, as it were. Whilst you were speaking, we ran a little poll of the audience in terms of, do we need an incentive for funding in terms of diagnostics? 100% yes. You mentioned treatments there, and obviously you're referring to the priority review voucher, which in itself, has, no real obligation for access as part of its structure, as it were. And I'm just wondering, we really need to, that really needs to be pushed onto the diagnostic setting, as it were. I know from speaking with Professor David Ridley and Professor Jeffrey Moe that they have worked on an iteration of the priority review voucher for the insecticide world to kind of catalyze novel research into novel insecticides. They call it the Verve. We actually interviewed them and put a three-way interview out with the IVCC and Professor David Ridley and Jeffrey Moe a while ago. And they, at the time, were actually working on a derivative of the priority review voucher for diagnostics. And I wonder, perhaps now, given the WHO's move, pushing for this kind of sovereign-led approach with funding there involved in that sovereign ownership, it might be time to try advocating for this particular type of funding mechanism, especially specifically for diagnostics. So I thought that last slide was really indicative of the reality behind behind diagnostics in terms of getting them out there. You mentioned some very interesting points there, and I think that's going to in itself raise quite a lot of coming here. I say thank you very much for that, Sarah. And don't worry about the sound. When it comes to the edit, we'll make it look very smooth. Don't worry. I've had worse things happen to me. Can you imagine if the background suddenly changes? I mean, some kind of Star Trek film because of the way these things work, but it was wonderful. So thank you very much for that. So we've heard from Beatrice in terms of the Merck's approach to this with access at the forefront in terms of consideration in all of their activities. We've heard from Sarah there in terms of Fine Diagnostics and their kind of approach to this, quite a technical half of a presentation, and then followed up with the access consideration that they uncovered in their 2019 study. Very interesting, both presentations and how they fit together. I think that leads us very nicely into the last the third presentation, Dr. Lieven Stuyver from Janssen. We hear a lot about the second shift in the roadmap in terms of a multi-sectorial collaboration, moving from disease state to a more wider look and bringing in different disciplines. Part of that process is bringing in different technologies and certainly different platforms and approaches. And I think this particular presentation really illustrate what's possible out there. It's very close to our hearts because, as I know, we have every year a meeting called D3, which is Drug Discovery, Development and Diagnostics. It's usually at the Wellcome Trust every year. And the last one, I believe it was in 2019, when way before COVID happened, the last physical event we put together. I know that Janssen and Merck had met there. And I think after that meeting, it really catalyzed the road towards this particular collaboration. So we're very, very proud and very happy of that. And we wanted to kind of just highlight that, as well as then obviously introduce the next speaker, Dr. Lieven Stuyver from Janssen. So I think over to you, Lieven. Would you want me to run your presentation there? Will it be easier because of the animation?
Yes, please do so. Okay,
no problem.
Okay, excellent. Thank you, Cameron, for the introduction, and Sarah and Bea for the excellent talks. It nicely set the scene for me. So today I'm going to talk about the A2STOP program. Maybe next. So I know it's a very lengthy and terrible title, but maybe we can stick for the introduction to this one single line. It's about affordable artificial intelligence, digital pathology for neglected tropical diseases. So if we would analyze this title, there are many components to this. So the first one is NTD. When it says NTD, it goes actually to all the different technologies that are using microscopy today for neglected tropical diseases. This can be stool-based diagnostics, blood-based diagnostics, skin-based diagnostics, bacteria and tissue, or even non-NTD use cases. So there are so many possibilities for this technology. The second one is on the, no, one second, yeah. So the second one is on the digital pathology. And digital pathology requires in a way that you have automated microscopy. Automated microscopy, that is a motor driving the slide, and that requires then an X, a Y, and a Z-axis motor. And especially the Z-axis motor, that's an important one because there you want to have the focus right on the object that you want to see. Having the right focus is extremely important when it comes to the artificial intelligence. And so in the artificial intelligence, that is the driver towards the diagnostic. And that is what is going to be compared to the standard of care when it goes to sensitivity, specificity, and so onwards. So for having the AI, the artificial intelligence, it needs to be based on, for example, an infrastructure, a database infrastructure that contains a large amount of images. And on those images, it needs to find the feature, in this case, for us, eggs of parasites. So when you look to this simple line, AIDP for NTD, there are many overhanging questions to it, a whole lot. But one of the most important one, that is, can you make it affordable? In the Western world, this type of technology is becoming more and more standard, and also it becomes regulated. There are, from the FDA, guidelines for software as a medical device that is specifically looking to these type of approaches. So for us, we would like to have a use case, and the use case is obviously connected to the J&J donation program for Vermox. So on the next slide, this one says that we are going to look to the donation program, and in the donation program, today it started in 2006 already. We have donated since 2006, and in 2015, 16, 17, there was the development of Vermox tubable, that is the mebendazole, which is now approved. And so there is a commitment of Johnson & Johnson to deliver 200 million doses annually up to 2025. Next to the donation program, it also says, and that's on the bottom line of the second, it says Johnson & Johnson is currently collaborating with partners to improve STH diagnostic capabilities. Now that says a lot, but what is it specifically when we talk about the STH diagnostic capabilities? For that, on the next slide, we have to look to the preventive chemotherapy intervention cycle. So this is extremely schematic, quite a lot of detail and associated with this, but typically it starts with a survey in the field. And in that survey, they're going to monitor for or measure for intensities of infections and the prevalence of infections. That information is paper-based and collected and then transferred in one way or another to a program manager who enters it into a healthcare system that goes to the Ministry of Health. From there onwards, if preventive chemotherapy is needed, can go to the WHO. From there, it goes to the donor. Donor is donating compound, and then the population at risk can be treated with the compound. So in the case of Johnson & Johnson, the compound that is donated is Vermox, and the amount of donation obviously is now 200 million doses annually, and we hope to treat the patients that are in need for this compound. But all of it and the request on a country base depends on what the survey is generating as information. So when Johnson & Johnson is saying we are going to look into innovation on the diagnostics, in a way, it is trying to improve that survey capacity. And there are many ways to do this. And so I'm going to explain today about one method, which is the artificial intelligence approach. In the next slide. So when we are talking about this innovation, we want to link it up into that donation program. And so there was last year from the WHO came that 2030 targets for soil transmitted helminth control programs. And in that document, they're talking about 6 ambitious goals. And the first one in the middle of the screen there, that is achieve and maintain elimination of STH morbidity in preschool-aged children and school-aged children. And that is indicated by the prevalence of moderate to heavy intensity infections of less than 2%. And at the bottom of that page, in that document, it says that the CATO-CATS is ideal for identifying those moderate and heavy intensity infections. So when we are now talking about new diagnostics or innovation, obviously there is already here on the 2030 targets, there is already some basis for it. In the next slide. So in that, for that target, CATO-CATS is now the standard of technology. I want to make just a sidestep. Beside CATO-CATS, obviously, like you've seen the presentation from Sarah on a rapid diagnostic test, there are many groups, including ourselves, that are looking for new biomarkers and new technologies to step away from stool and go to non-stool-based testing. However, based on the information that we have collected in our lab, that is a very complex discovery process. And to our understanding today, the cost and the timelines for the development, the multiplexing complexity, because we are talking about 4 worm species, and then all the discovery and development that is needed, it's our belief today that those new biomarkers will not become available to inform the 2030 roadmap of the WHO. And so, therefore, I think it's important that we understand what the CATO-CATS method can bring to the field. Just one step back, the CATO-CATS method is a very old method. It was first published by Cato in 1954. I have never read that article. And then there were a few additions to it, but finally CATS added the final component to this, and that is published in 1972. And so since 1972, this method has been largely unchanged. So it's more than 50 years that this has been used as a routine procedure. In the next slide. Yeah. So we have conducted a market research, and that was executed by Health Advances. And I was asking the end users and the customers on the details on the CATO-CATS. What is their opinion and what is their appreciation of all of it, and where are the pain points? And so the information that came back to us, that is the highest level of the unmet need from the market research, is on the slide evaluation. And there are a few other components, like slide preparation obviously remains difficult, but then look to the data capture and the revise of the MDA. So that is the bucket that is the most complex. And the reason why the slide evaluation is mentioned there, that is, it requires a lot of hand-on time. It has limited sensitivity and reproducibility, and you need to have a well-trained microscopist to do that. So there's quite some possibility here to improve it. Now, just to point towards it, Specificity of the technology was not mentioned in this market research, but they came back to sensitivity a few times. And so sensitivity in the current keto-CATS method, and it was also mentioned in the previous talk, the sensitivity is something that people are looking to, and especially in the low prevalence settings, that becomes a difficult position. So in the next slide, when I was researching artificial intelligence and digital pathology in the literature, I found this statement. So it says, although humans are really good at making visual and informational associations, computers are better at finding the proverbial needle in the haystack and being relentlessly consistent and rigorous. So if you would apply this statement to the keto-CATS method and to the topic also of sensitivity and on the reproducibility, I think it says a lot on why artificial intelligence and digital pathology can bring improvements into this NTD space. Next slide. So I'm embarking on an affordable AIDP concept. You need partners for it, especially because there are multi-use cases in NTDs. And all of these partners that you're working with will bring their own expertise and resources that you need throughout the process of R&D and access. In our case, we selected the use case on parasite detection in egg, and since schistosoma eggs, schistoma soni eggs, and also STH eggs are found in stool, yeah, this technology can inform both the donation program of Vermox, but also the donation program of praziquantel. So in a way, it's logic that Johnson & Johnson and Merck are working together on this innovation. So in July 2020, both companies forged such a unique first-of-its-kind industry partnership. And in a way, it is also building further on the commitments that were made in the London Declaration. Next. Besides the collaboration and that we have with Merck, we need other partners. And we built that consortium in which we are trying to bring the whole concept to the next level. In that group of partners, first of all, we have Etteplan. That's the engineering group in Sweden, and they are building the prototypes. We need academic partners to go through the complete thought process. And we work with Ghent University and also Jima University. And those are actually two labs with extensive expertise in the technology evaluations for this use case. Since we are aiming to bring the technology in a head-to-head comparison to the standard way of testing, we want to be associated to programs in the field. And so we have partners in APHI and also in Ethiopia, and also in the Vector Control Division in the Ministry of Health of Uganda. And finally, we have ligature and bridges to development, and they play a crucial role in the communication with the community and the stakeholders, but also for the adoption preparedness. There's quite a lot of additional needed beyond the science and the technology. In the next slide. So as mentioned before, the consortium is aiming to innovate the slide evaluation, the data capture, and the revision of the MDA system. So the technology that we want to introduce, that is automated slide scanning, the whole slide imaging and artificial intelligence, and that combining it with a dashboard that is in real time almost available for making the decisions for MDA. So that is the component that we are focused on. On the next slide shows you the workflow. It's a proposal for the research components that we are doing now, so it's not yet written in stone. It's open for further evaluation and discussion. But it starts with digitalizing the field studies, so stepping away from the paper workflow. And once the study is entered into a digital system, that information can be accessed by the control unit. And that's the unit that you see there in the middle. In fact, that is a computer that has data storage capacity and an AI processing capacity. And that control unit will allow barcode printers to print the barcodes, which can go on the stool pods and on the slides and so on, and on the paper if there's still paper. But it will also control the slide scanners to start scanning, to collect the images and send the images back to the control unit. The control unit will then make the whole slide image, run the AI on that whole slide image, and that report that is then generated, that it can be sent through the healthcare system as a simple data package. So you don't need large data information or images to be sent through the internet. And so the difference of this approach is that we, in a way, don't need internet. It can be operating in a remote area without internet. But in cases that internet is available, it might also communicate to an A2stop server, which can push new artificial intelligence code to the control unit or pull information of the performance of the unit back into the cloud server. So the image that you see there at the bottom right, that is the current prototype that was engineered in Sweden, and I would that's the scanner, and I just would like you to show on the next slide. That is the whole slide image there, and you can see here it's a digital image, and if you zoom in into that image, you can start seeing the eggs of Ascaris and Trichuris. So looks like an image, a whole slide image with sufficient amount of precision and accuracy for the artificial intelligence to start looking for eggs. This is a slide that was from Ethiopia. It's a little bit a slide that is by now almost 2 years old, but we are looking for new slides, and we will further evaluate the technology on new slides. Besides the whole slide imaging, there is a component of artificial intelligence, and that is on the right-hand side here. There is a graphical user interface that is generated, and since this is a schistosoma meeting, so I selected here a schistosoma egg that was recognized by the artificial intelligence in a stool sample. And I don't know if you can read it, but confidence of the AI on this feature detection is 99.7%. So you can be pretty sure that this is a schistosoma egg. So the artificial intelligence as it is today, we built that on information that was collected in a library in total on images of 17,000 eggs. And when we perform this artificial intelligence on unseen data, today we have an analytical sensitivity and specificity that is greater than 95% for all the species that we tested. Okay, so we are pretty confident that with these technologies as they stand today, that we have totally de-risked the part on the whole slide imaging and also on the artificial intelligence. On the next slide. Yes, so on the next slide, it tells you more or less where we are today. We are still in research, and we are designing prototypes. The prototype that you see there, that is a picture of prototype 2, At the moment, in the Etteplan group is now working on prototype 3, next generation. So we will continue to work and improve on those prototypes. And for the rest of the year, we will see what the performance of those prototypes are against the target product profile that is available from the LIM paper 2018. And so that is going to be the main objective for the remaining of this year. Performance testing and eventually comparing it to the standard of testing. When will we go into a design freeze and start producing beta prototypes? That depends on so many factors. We don't know yet, but it's not going to be this year. That will be somewhere in next year. When we build prototypes, it's not a prototype only on the scanner, it's prototype on the control unit, and might be prototyping the whole system as it is given there in the picture. And then the development can take some time, and then we will need, obviously at the end, we will need to bring the technology to the field. And as of today, that is maybe the biggest headache that I have and that many people with me have. That is, and also in the previous talk, Sarah in her last slide nicely summarized it. So how are we going to bring artificial intelligence, digital pathology for NTD into the field in an affordable way? There are quite a lot of questions around it. And so with this audience, maybe we can also start additional thinking on the way for doing that. All right, so the next slide is then my conclusion. So you need, if for ambitious projects like this one or the previous one, it's critical to have strong collaborations. And it's just the strong collaborations and all the perspective of the different groups that is driving the innovation. This technology is actually a second generation CALTO-CATS. It's building on a 50-year-old technology, and it's taking it to the next level. And it's an opportunity to improve the STH and the schisto programmatic decision making. So when we were presenting this to the community so far, we have only received extremely positive feedback. There are a few questions on affordability, and there are questions on the access component, but in general, it's been very, very positive. As mentioned before, affordability and scalable, those are the key questions, and we need much more inputs on how we're going to do this. And then finally, so you have seen we are building hardware and software for one use case within NTDs. But that is not where it should stop. You can add many more use cases within the NTDs that need microscopy, and you can even go to non-NTD use cases. So the success of this type of technology largely depends on the menu, and I can envision a menu list of use cases for this technology, but obviously it's dependent on partners, on investment, and on vision to bring it forward. And with that, I think I would just like to, next slide, Cameron, if you can. Yeah, so again here, we are working with different partners in the field, in the industry, especially the group here in Etteplan has been extremely useful for bringing innovation both on engineering and artificial intelligence. But we need the partners also for technology evaluations like Ghent University and Jimma University, the partners for evaluating it, EPHI and the Vector Control Division, and partners for management and engagement, bridges to development and ligature. The images that we collected, they were actually taken within the Starworms program, and that was done together with a whole group of individuals that are listed there on the right-hand side, in Kemri, in Kenya, in Cambodia, in Tanzania, and in Ethiopia. We are very thankful to the people for helping us and to prototyping this technology. And with that, Cameron, I would like to give it back to you and thanks for the opportunity to present this innovation to you. Thank you.
What can I say? I mean, that was absolutely amazing. Not just amazing. People think about AI. They think about, I guess, you know, bioinformatics, data mining at the early discovery stages, protein folding, these type of applications, and yet to see it in that sense is absolutely really staggering in terms of where that could lead to. And you mentioned some of these things in terms of affordability and scalability and partnerships, the importance of that. I'm just wondering what type of partnerships are going to develop on the back of that kind of approach. Absolutely visionary stuff. I remember we had a phone call with you about 1.5 years ago when you were just getting into this, and even at that stage, it was so such a seminal moment, really, that, oh my God, this is it. This is an amazing vision that they've got, and to see it now to where you've got to via this collaboration with Merck, I think it's absolutely amazing, and certainly some form of blueprint for moving from rhetorical positions on multidisciplinary, cross-sector. We've heard that for years and years and years. This could well become a blueprint in terms of moving forward. Honestly, thank you very, very much. That was amazing. And some of the comments coming through are absolutely, you've won a lot of people over there. So amazing, amazing. We've been joined by the NIH from the US. We've got J&J have joined as well. We've got people coming in from all over the world, actually, on this particular one. Dr. Nebe from Nigeria in terms of their Schisto program and their STH program. All sorts of countries and people joining in. I wish I had time to give everybody a shout out, but it's been really an amazing presentation. I think that takes us to the last presentation, which is back to Dr. Beatrice Greco, the head of R&D at the Merck Global Health Institute, because as an effect, you bring everything back into impact towards access. I know we scheduled this until 3:30, but if everybody's all right, I was going to ask this question. We're going to go a little bit over. Sarah, I know you have to go. Is that correct? If it is.
Yes, I'm really sorry. I really would want to be part of the Q&A, but I apologize for this. But I have to go. Apologies.
Absolutely no problem. You mentioned that before to us. You had a hard stop. I'd just like to say thank you very, very, very much for that. That last slide you showed really kind of brought it all back into the reality of access and the reality of pushing forward on the diagnostics front. So much work to do. And I'm sure the Q&A will relay it back to you, and certainly the edit will show that. And I hope we'll have the chance to pursue this. We're certainly looking to build a continuum around access in the coming months. So I'm sure we'll talk again. But thank you very, very much for that. So,
thank you for having me. And if there are any specific questions, about the work, I'm very happy to take those questions and respond to those who have asked those questions. And I'm very happy to do that after the talk as well.
Thank you.
Thank you for having me. Thank you.
Keep safe. Bye-bye.
Thank you. Bye-bye.
Thanks a lot. And Beatrice and Lieven, are you okay if we continue? And I guess I'm asking that same question to the audience as well. Are you both okay if we continue a little bit?
Sure. I think I will have, when we have the time to go through the last presentation, because I would really like to bring all those topics of access a little bit together now. And we may not have necessarily the time for Q&A session, but as Sarah was saying, maybe we can collect all the comments and then through maybe your platform, share back our answers to whoever has raised a question.
That's a great idea. I think there's so much to unpack that this deserves a session just on Q&A. I think you guys, you've blueprinted how to collaborate, and this whole AI angle, Lieven, you guys have lit a fire. I think a lot of people are thinking, so I'm going to come off and I'll leave you guys I'll leave you, Beatrice. So yeah, over to you, Beatrice. All right, thank you. Sorry.
Thank you very much. Okay. I think what I'd like to do now is to summarize or to bring together a little bit of our thinking and what we've been observing by working both from the drug as well as the diagnostic angle. And you heard from Sarah as well as Lieven that already from a diagnostic perspective, there's really a lot of pending questions associated to access of those upcoming innovation. And I can share also that from the pediatric praziquantel perspective, similar elements are there, and it comes to the point where we really need to start thinking about creating an access path for those innovations. So, but first, and I believe most of you are familiar with the 4A frameworks. What are the key elements that are framing an access path? As Sarah was alluding, as well as Lieven, there's some elements about affordability, etc. But in a much more structured manner, if we look at this framework, it starts with availability. Availability means that there is the right manufacturing and manufacturer interested in producing that particular product, associated with the right forecasting, with together a supplying and ordering mechanism. This is associated with affordability. Affordability, as Lieven was clearly showing, it has to be thought through as early as research to make sure that at the end we have a product when it's launched that is at an extremely affordable cost. But it's not just about that affordability. Affordability is also the mechanism of procurement allowing to buy from the manufacturers and to allow the countries to have access to these innovations at prices that are quote unquote affordable. And affordability is also about looking at the landscape of donors, insurance, out-of-pocket payments from patients, and seeing how this can be organized. Then from an acceptability perspective, this is the third element, acceptability at the level of health authorities, but also at the level of regulatory bodies in a local level and regional level. And that was also one element that was alluded by Sarah, talking about the fragmentation and the way to strengthen those frameworks that are needed. The regulatory bodies there, but it's acceptability also at the level of patient and the medical bodies. Then the appropriate use, this is really how this is going to be implemented locally, how is it going to be used in terms of not only for the drugs dosage and management of side effects, but the proper implementation and maintenance. But this overall framework, this architecture, which is almost the 5th A of the framework, comes hand in hand with, in the background, the incentives, the financial mechanisms that is maintaining that framework working together. So there is the need to have really that financing aspect behind it. When we look now at, based on our experience with now drugs and diagnostics, those upcoming schistosomiasis medical products, and we look at this framework, we see that actually there are some elements where there's some gaps. It needs to be reinforced, the acceptability, and we talked about touching upon the regulatory framework that would need to be strengthened, in particular for diagnostics, or at least better understood. There's some artificial intelligence also here from a regulatory perspective. How are we going to tackle this? But there are some elements that are already existing from acceptability, appropriate use, that need to be adapted for the innovations that are coming. And this is actually some of the work that, again, we are doing with the pediatric praziquantel formulation access path and the ADOPT program. But beyond those two pillars, if we look at the overarching framework of access, well, we do see that there are some very critical gaps in the availability. We see that the manufacturers are reluctant to walk into this game, to produce those type of innovations. In particular, we are thinking about in the frame of moving slightly away from from a drug perspective, from donation to more sustainable access. Similarly, for diagnostic, it has to be a sustainability element into it. So manufacturers are at the beginning of the story. And today there's very limited interest from manufacturers because from a market perspective, there's really an affordability push towards low cost, and therefore there's limited incentives for them to get into that game. So manufacturing is an issue. Forecasting is today not existing, really. It has to be built, it has to be created, as well as the supplying and ordering aspect. When it comes to affordability, as I was saying, it's beyond the work that is done within the R&D space. It's really about, do we have today, for schistosomiasis innovation product, a procurement mechanism that is going to ensure that countries are going to have access to those medical products? No, there's no such a thing as a global fund for NTDs, a global fund for schisto innovations that we're preparing. So here there's a major gap, a major element on which we need to work. Similarly, the other element that is sorely missing, it's the financing backup of all this framework. Today it's very fragmented. The funds that are available are either dedicated for R&D per se or for implementation and some aspect of implementation locally. But in between, there is really a lack of financing mechanisms and funds to maintain that framework working together. So this is really something that we have to create, we have to coordinate. And today, we are already exploring some of those elements, in particular in the context of the pediatric praziquantel, but also in the context of the rapid diagnostic test we are working with FindWell. And in terms of availability, for example, for us, one element and one way of creating incentives for manufacturers to be part of this story is to foster local manufacturing capacity through technology transfer into some of our African partner countries. The forecasting and ordering, this is about engaging dialogue as early as now with relevant international and governmental organizations to create this future demand, but even just the mechanism of having forecast. The affordability, and again here it's a key element of how do we create a procurement mechanism. Today, all these activities of affordability financing mechanisms are really at the level still of advocacy, discussing with stakeholders, investing into also creating an investment case to ensure that when we go and talk with those stakeholders, we do show the benefits and the potential impact of those innovations in the context, of course, of schistosomiasis. And with this, I just wanted to end with the last slide, which is really here a call for action to the community here. When we look at drugs and diagnostics together for one given and neglected tropical disease, and this is the case for schisto for us, what we see is that there are very general, similar issues associated with the various elements of the access framework. And in fact, the issues are leading to very similar also potential options and solutions. When it comes to availability, there's a clear need to look at incentivizing manufacturers to ensure that they generate the right capacities for both drugs and diagnostics. So here there's a need to think as a holistic approach rather than one product versus another one. Again, aligning the forecast for the NTD products and here for the schisto product would be a way of also leveraging resources. When it comes to affordability, there are talks about looking at pooled procurement mechanism, but also creating more global NTD funding platforms in such a way that we are addressing more than one element at a time and much more holistic approach for the countries. Similarly, for acceptability, appropriate use, they are very similar type of solutions. And when it comes to, and Cameron, you alluded to it with the PRV, when it comes to incentives, there's indeed PRVs for incentives of R&D, but today there's really no incentives for access per se. And here we strongly believe that it's time now for this community to really think about a global access framework for NTD products at large, if we are serious about reaching elimination of a number of them by 2030. So with this, I just thank you for your attention, and back to you, Cameron.
Beatrice, thank you very, very much for that. I think that really tied everything together and brought us back to this whole access framework. We can see, well, from the kind of technical presentations, kind of whereabouts it's touching, whereabouts this kind of approach would touch on the healthcare systems, as it were, and what kind of partnerships are kind of needed really moving forward. I think that the funding issue is going to be massive, and perhaps that's something that's going to be answered by this kind of country ownership shift in the WHO map, i.e. much more of a sovereign look. We already have a fragmented regulatory framework out there in terms of the need to harmonize on that basis, but also from a funding perspective, maybe countries need to come into this with a totally different investment kind of mindset and look to bring this forward a bit. And I think definitely the time is now to start advocating on that. I'm just very, very, very wary of the time. A lot of people are starting to drop off in terms of the attendees. The feedback is absolutely stunning. People are absolutely, Daniela Fusco, thanks Dr. Beatrice, is very, very enlightening. We've had people already asking for partnerships. So Josephine Gunjiri kindly forward us the opening for collaboration on FGS in Kenya with emails. We've had other people as well. So we've had Laura Brown, who works with the WISER project, the Michael Templeton's group, asking about what is the kind of size of databases of eggs that you're looking for and issues around that. Guilio de Leo is asking what is the size of the training data sets, what deep learning algorithms are you using. People are really engaging around this idea. So it's not just this is way beyond the donor collaboration, but it just shows when two heavy organizations that are heavily committed to this do something, the ripples of it through the kind of space we're all in really are, it really does something, and it's going to really catalyze a lot of movement towards you. So I'm unsure what to do. Should we continue? They're all going to say yes, but then I'm worried that some people are a lot of people have dropped off. You obviously very busy people as well. So a suggestion is all the questions we've got loads. If you're okay with that, we could put those forward to you in written form and then perhaps formulate another session where we take those questions and open up those channels with a view to bringing in a bit of more of a partnership feel to it and a bit more of a tangible, new partners perhaps into this or areas. Is that something that would work for you?
I think it would be fine, because I think right now, unfortunately, the time is very. But yeah, let's collect. We will be happy to answer, as Sarah was saying, we'll be happy to answer a lot of those questions, and we look forward anyway to continue this conversation. Yeah. Again, to your point, Cameron, that first session was really to share with the community some of the work that we do, but not only that, it's really, again, and this is the example of having Lieven here, it's to say, okay, what kind of partnerships do we need to think? Because I truly believe that R&D, it's not that it's well taken care of. We need to continue to invest into it. That is not a question. But innovations are going to come and to be launched within the next couple of years. If we are not now preparing the ground for the access, this is going to be a waste of resource, a waste of time. It's going to be a big failure for the entire community. So my call here is to say we need to move the speed up from great work is being done from an advocacy perspective, engaging stakeholders, discussions. We need now to unlock some of those various elements that we are highlighting in the last presentation, because they are essential to make success, not only from the schisto perspective, but from the soil-transmitted helminth aspect, from other NTDs. And I think this is really there, this call for action from the community. We need to have a global access framework. Yeah. It's just about framework, but trickling down into very concrete actions now.
Yeah. I think you hit it on the head there in terms of what's needed. Lieven mentioned the term de-risking collaboration in your presentation. De-risking access by including a wide array of partners in a granular feel to the voices involved in defining and redefining access. I think that's definitely the way forward. And I think that's going to have its own ramifications in terms of the money, the capital flow around investments in this. If the people of those areas the local voices are involved, it makes for a far more pressing case to change political will in terms of opening up investment in this area, these harmonization issues on the regulatory landscapes, all of it. If they feel that they are involved, they have a stake, they have a place at the table. So very, very interesting to see how this evolves. And I think the way you framed it, the workflows and stuff in terms of the AI side of things, there are so many points of contact where the local researcher can really get involved. I think that's reflecting in some of the questions that are coming through as well about the databases, about this kind of stuff. So, hats off to both of you for that. That was absolutely wonderful. And this really is a blueprint. So, anyone out there, we're going to get it edited and kind of, it'll run in a smooth way. But I just wanted to say thank you very, very much to both of you for making time. I think I think this is a good place to stop, because it's going to we're going to get in trouble with a lot of people in terms of. Thank you so very, very much. Yeah, really. All right.
Thank you also for having us.
Thank you. Absolute pleasure. And we look forward to the next one. All right. Yeah.
Thanks.
Thanks. Bye-bye. Take care. Bye. Ciao, ciao. Keep safe. And thank you, everybody, for joining us. Take care, everybody. Yeah. Thank you all. Keep safe. Bye. Keep safe. Thanks.